Cool. Yeah, last seminar of the year. So first of all, yeah, let me start by saying thank you to all of the organizers who did great organizing. It's been a cool year of seminars so far. And thanks to the people who signed up for next year, which is also shaping up to be a nice set. Yeah, this is, so last seminar of the year, and it is the last seminar of the Digital Approaches to HPS series. And so we ran out of people to invite who were cheap, so we invited ourselves. So we're just going to talk a little bit about some of the results from
it was the end.
latest
It
project.
was a fabulous
The summation of,
yeah,
no, it's just, yeah, we're out of money, so we're free, which is cool. But yeah, we're going to talk a little bit about some of the new data. We've been doing some fun stuff, and we think we like it. And so a little bit of methodology and a little bit of results from this project on trying to understand the taxonomy literature with a bit of a focus, as you'll see, on trying to figure out why everyone is yelling at each other all the time, which seems to be a real pastime in taxonomy. Lots of collaborators to thank, especially, I know Marlies has worked a bunch in particular on the classification stuff. I don't know if you want to say something about other contributions, but yeah, we've worked with a bunch of people on this.
Yeah, so there was a lot of labeling of like pieces of text, and the taxonomy set helped reading a lot of like insect body parts and stuff like that we don't understand.
Yeah, much appreciated. So yeah, what we're going to do, we'll talk a little bit about what the data is and how we got it, what we did, a bit about what we kind of pulled out of it, what we were trying to classify and understand, tax allocations, topics, disagreement, and methodology. So the method stuff is kind of the taxonomic methodology stuff is the one part of this that's actually cool enough that we're thinking about publishing an article just about the method that we did, because that's actually kind of neat and interesting. It's by far the most interesting use for an LLM that I've found so far in doing philosophy, so that's kind of fun. And then we're talking about what we think we might be able to learn with what we get from that data. Some stuff about, again, methodology, but then also where do people do taxonomy, both north-south and also in the sense of in the environment, and then again about disagreement more specifically. So here's what we have. We have about 43,000 journal articles in various areas of taxonomy. That's a quick rundown. The two features that I always mention when I talk about this list of journals, so if you're asking why these journals, imagining you're an angry peer reviewer, the two guiding features that pick these journals out, one of them is these are journals where almost everything in them is nitty-gritty nuts and bolts taxonomy. It's where you go if you want to describe a new species, right? They don't have general biology in them. That's not the kinds of publications that these are. They also, and this was very on purpose, mostly don't have fossil taxonomy. They mostly don't have any paleo stuff because different ball game, right? Different rules of the game for describing fossil species, and so we tried to avoid them on purpose. The downside of that is that they're pretty new. Sorry, these axis labels are like garbage. the scientists would shank me for this. That's 2000 where it picks up off the x-axis in the middle down there, though. The right-hand cutoff is just when we quit downloading papers. But it's 2000 when it really takes off. I mean, not super unsurprising. Before that, there were different journals. I mean, a lot of the development of this collection of journals is around kind of a realization by the taxonomic community that we can do internet published stuff in a different way for describing species, and so there was a shift. If you wanted to get older data, you'd have to do stuff like go get big taxonomic treatment books and figure out how to break them apart, which would be a mess. So we're also trying to kind of limit ourselves here. It doesn't mean that the quality of the data, in comparison to the DH stuff that I normally work with or talk about, the quality of the data is, I think, is actually much better because it's reasonably recent, like nice PDFs, right? It's not like ugly scans of stuff from JSTOR. So that helps. And even in some cases, it's actually nice, like XML markup, like text-based source files, which is much better. Okay, so that's a bit about what the corpus is. I could have put in some more slides. I didn't bother adding them here. It's decently representative across the different groups in the tree of life. It's not perfect. There's a few. Mostly, yeah, almost exclusively. There's a few. I've seen other languages bubble across a couple of times, but I think it's pretty rare. I think it's a small handful. What can we do with it? What can we analyze inside here? So the first and most important thing you might want to know is, well, what species are they talking about? And we are actually very lucky here. We can quite simply lean on a delightful software package that is, in part, actually, this is very cool. In part, I think it's in some way connected to the Biodiversity Heritage Library. They use it a lot for analyzing BHL texts, which is this great online resource of historical texts in biology. Really cool project that simultaneously, it uses a combination of looking through the text for things that look like they should be species names and matching against a dictionary of things that it knows are species names. And then once it finds plausible hits, it actually goes out and confirms them. This will be important, this will be useful later for some of the other analyses we want to do. It goes out and confirms them in some of the big databases that summarize the known species on Earth. So stuff like the GBIF or the Open Tree of Life. You can link this to different data sets. And so when you have one of those hits, you can be pretty sure that like the paper actually talked about that species. It also matches higher groups, which is useful sometimes too. So if there are mentions of families or classes or whatever, it also catches those. So that's really nice, and it's frighteningly fast. So that's not even hard. Something that is a little bit harder. It is a language model, although it's not really an LLM. It's a medium LM. For another thing you might want to do, an obvious thing to want to analyze is, where did the work happen? Where on Earth are the species that are being studied in a given paper? That's a really interesting and challenging computer science question. I've used now in the 20 years I've been doing digital humanities, I've used 3 or 4 different pieces of software to do this. This is the current state of the art, which is a medium language model, 334 million parameters, so it's pretty small. That's trained on a data set of, really what it is they call it token classification. So it classifies place names, but it also classifies people names, organization names, company names. They used to call this named entity recognition, but that's the old way of doing it. Now we use this. Thankfully, yeah, the state library of Bavaria decided to train me a really nice model for this. Thank you, Bayerische Staatsbibliothek, which is the current kind of pack leader. Does really, really well in English language text at finding sort of, understanding where in a sentence the indication of a location name would normally be and flagging that. That's cool. Now you have location names out of the text, but that's not actually useful because you can't analyze those because they're just text. What you can analyze are latitude and longitude coordinates. So how do you get from hunks of text to latitude and longitude coordinates? One thing you can do is you can pay Google like tens of thousands of euros and have Google do it for you. This is notably what Google does when you go to Google Maps, right? And type like whatever, the name of some random park, right? It has to scan that and turn it back into a latitude and longitude coordinate to put the pin on the map. Google's very good at it. I don't have enough money to pay Google to do it. What you can do instead is you can go out and get a list that is commonly called a gazette or a gazetteer, that is thousands and hundreds of thousands, millions, I think even. Yes, it's dozens of millions of names of places around the world and corresponding latitude, longitude points, and then just do comparisons. And that's not as good as reverse geocoding. I mean, I've run tests about this, but at least it's going to get you close, right? So it may not have, Google is going to know the name of like every wildlife preserve on the face of planet Earth and be able to map them exactly. That this thing isn't. But at least if you talk about some wildlife preserve in Thailand, you're also going to use the word Thailand, and we're going to match the word Thailand, right? So at least we're going to get in the region. So that's what we do. This is not fast. This took my laptop, or not my laptop, this took the desktop up there in my office like a month. You crunch through every single place name. First you look for an unambiguous match in the database of hundreds of millions of other place names. Then you look for fuzzy matches. You try to rank the quality of the fuzzy match into the database. Eventually, if all else fails, what we do is we sort of give the paper a geographic center. So you take the average of all the things we know how to identify, you put that point on the map, and then you see if the other point is closest to it. Hoping maybe that like if you did most of your work in India, then like if there's another point, it should probably be in India. This is a mess and it's very hard, but it does seem to give pretty reasonable results in the end. It's not spectacular, but it works well enough to get the data off the ground.
Charles,
Yeah.
question for clarification. So what places are you interested in here? Like, I guess where the species is living or is found, or
So-
like how do you distinguish the lab where it's research?
Good. So what we hope to be doing, and this is actually, I need to clean this data up and I haven't done it yet, so what you're going to see today is a bit out of date. Of course, in the text of the articles will also be things like, Université Catholique de Louvain, which is probably also in the place list and therefore also gets a pin on the map, right?
Yeah.
We try to cut that stuff out and just look at the body of the paper. And then really we'll come back to this in a little bit when we talk about this north-south question. The proxy variable, the proxy that we're trying, that this is trying to get, the variable for which this is a proxy is something like, where in the world are taxonomists paying attention to? Like that's the feature about this literature that is really interesting. And taxonomists have for a long time, we'll come back to that just in 5 seconds. Taxonomists have for a long time been worried that they're not sort of correctly distributing their attention across the earth, right? And so partly that's where the species lives. That's also where, the field work happened, whatever it may be. The places that they talk about are presumably the places they're paying attention to.
Thanks.
Now, I've talked about this before in CEFISES seminars, although this time I'm going to talk about it much less because it was less useful. We also tried topic modeling on this data set, which is the standard way to sort of classify documents based on their content in a kind of unstructured way. You don't train the computer in advance. You just say, hey, computer, what are these papers about? Here, this worked much less well, unfortunately. It very often is picking out topics that are actually essentially just proxying for species. Because, unsurprisingly, right, if you work on, I don't know, mollusks, you have a very specific vocabulary that you use when you talk about how to describe them of all the body parts that you need to measure to know what kind you have. And so that's going to be the dominant signal. And so most of the topics in the topic model, well, if you're just going to talk about species, you may as well talk about species. We already extracted the names of the species. So this is an indirect way to get to the species. It's not that useful. A few might be okay. There's one here that looks like it's maybe about DNA barcoding. But I'm not sure yet whether there's a useful sort of result in the topic models in this data set. We'll find out. We'll see what we can learn. Lastly, something we've been worried about on the project for quite a while is, yeah, as I mentioned briefly, it does seem like taxonomists spend a lot of time disagreeing with one another. It's a kind of well-known, it's a thing that scientists really like to complain about, at least, that taxonomists especially. I was giving a version of this talk when I was in Georgia, and I had one of the leading ecologists of the Caucasus in the front row, and I said something about this, and he went, "Oh my God!" It's like, yeah, you know, it is known. It is known among practitioners that there is a lot of fighting. It'd be cool to be able to measure that, and then if you measure it, you can look at what it correlates with, and if you see what it correlates with, maybe you can start to think about causal explanations, right? That would be cool. So painstakingly, Stein sat down with a bunch of articles where we knew people were fighting, and tried to get to lists of terms that sort of are the kinds of words you would use when you're having an argument in the literature. And we made 3 lists. There's a list of just disagreement words. There's a list of kind of epistemic value evaluation words. There's a list of just pejorative words, like just when they just insult each other, because they actually do that a lot too. Actually, it turns out that the 3 of those, there was a 4th list too that didn't really work, but the 3 of those in most of our experience are fairly decently correlated, which is kind of interesting by itself. What you could do then though is, and I mean this is just like a dead easy, simple, stupid analysis. Literally just look at the relative frequency of the terms on that list in all the papers in the database, and then you sort of give each paper a like disagreeability index. Where it goes from 1.0, which is, you know, probably some kind of small corrective paper, right? That's very short and loaded with these terms, all the way back to 0. And then that's a quantitative variable that we can use for analysis in a little bit. Okay, let's pass it off.
Yeah, thanks. So the final data assembly thing I'll talk about is the methods, which, as Charles said, is sort of the crown jewel of the project.
The cool new thing.
Cool new thing. Also, I think just because methods are an interesting thing in science, like I know philosophers have had a bit of an obsession with methods always, but that was the general thing. Like what we're trying is to look empirically in detail what they're actually doing, which I think will say a lot about the kinds of research they are doing. But it's quite difficult, I think, probably in every field, but particularly in taxonomy. Because there's no standard like references or software packages or anything like that at all. Like in some fields, you have this particular software you use for the analysis, or everyone cites this particular paper. but they don't do that, so there's like no shortcut to the methods. Another thing which will come back a lot is that there's a lot of different research communities. Like every taxon, like slightly higher level taxon, has its own journal, its own society, its own tradition that goes back 2.5 centuries, its own collection, its own amateurs. And they have their own terminology, they have their own methods, they cite each other, they don't really look at other research, so they're all very isolated. And so it's very difficult to track methods across these things. They use things that are similar, but they use different words. Then another thing that is very interesting about taxonomy, I think, is that it's a very exploratory science. So it's not like other fields that have hypotheses and they test them. It's more like you go on a field trip, you look around and you find something, and then you describe it. So we wrote a paper about pre-registration in taxonomy where we try to argue that people should try to do this, but the taxonomists say, like, how can we do this? There's this infinitely large space of variation and we don't know what we're going to find, so how can we say how we're going to find it? So there's no tradition at all of reporting on your methodology. I think maybe 60% of the papers had a methodology section. Most of them had like 2 paragraphs, 1 paragraph, where they say the specimen is stored in this museum or something like that. So very little information. The other thing is that taxonomy is like 50% amateurs, which is nice. It's a lot of labor you can use, but they have different standards of reporting. They use less complicated methods. They don't have the funding and the resources. So that means that within taxonomy there's often 2 different streams of research happening. There's like the university level research, and then there's the amateurs that go out with their club and find something and write very small papers. It's also interesting to look at our corpus because, as I said, taxonomy is very old. It's this hobby of rich English gentlemen from the 18th century, which remained quite as it was until the late 1990s, and then molecular methods arrived, which many people think have changed taxonomy dramatically. So it's interesting to see to what extent that's actually the case. And I think it will be reflected in the methods, because the molecular stuff is really the methodology. So to extract the methods, first thing we try doing is making some kind of high-level classification. So we're not interested in the really fine-grained stuff. I think we ended up with something like 40 categories, which is already quite a lot for a classifier. So I sat together with a couple of taxonomists and we first made a very broad structure like molecular methods, phenotypic methods, and then like data processing, data analysis, data collection, just to have broad categories. Then we isolated the methods sections of our papers and did some exploratory analysis on it. The kinds of topic models that Charles does as well. Here they were quite useful because it was only the methodology sections, and we really got a lot of topics that were clearly about like PCA or DNA sequencing or stuff like that. So we went through them, we used the biggest ones, we added some expert domain knowledge from the taxonomists to get a rough classification, and then we just started annotating random paragraphs at first, just randomly sampled. We did this a bunch, then designed the first model, just a very simple classifier, used that classifier to see what samples it was most uncertain about, and then used that to do some targeted sampling of more paragraphs to then annotate so we could give some more information to our classifier. And then we also selected some paragraphs that we thought would be from labels that were very sparse. So some of the things are very common, some are very sparse, so we selected for that.
Yeah, just a clarification question. Annotation means manual annotation.
Yeah, so people, so it was me and then 4 taxonomists. In the end we did around 1800. which was a lot of hours of work, but it's very little for machine learning. So the big bottleneck in this whole project is the data. So then once we had that, we trained a couple of classifiers. I'll talk more about them in a second. And then just for comparison, we also used GPT 04 mini and some 4.0 through the API to see how well it would compare. The LLM was just zero shot, so we didn't give it any examples. We just gave the bit of text, we gave the question, and then just do the answer. So very different from the other classifiers that got the 1800 samples. The LLM didn't get anything at all. So that's the classification we used in the end. The green ones are the phenotype-based ones, the purple ones are the molecular methods, and then there was some miscellaneous stuff that we didn't want to ditch, but it also didn't really fit in any of the other things. I don't know what the random is actually. Maybe that's just left over from the... The figure was made by Claude, the model actually, the LLM, just on the base of the prompt. I think it looks really great. But the random is some hallucination, I think.
What does interbreeding mean as method?
So one of the main ways in which taxonomists distinguish species is by checking whether they can interbreed or whether they do actually exchange genes. They do this both with phenotype-based methods and molecular methods, and it's quite interesting to see to what extent they do, because it's like a different paradigm about what a species is. If you look at the interbreeding or you just look at the phylogeny.
In parentheses, because it's a well-known philosophical problem, you might also hope that they would tell you what concept of species they were going to use. Despite the fact that, for several of these journals, it's in the instructions for authors, right? That you are literally supposed to put, if you're defining a new species, you're supposed to put what species concept you use. Of those 44,000 articles, something like 600 make reference to any concept of species whatsoever. So that's not going to help either. For a while we thought, oh yeah, that'll be a variable we can use, to help and no, that's the n is too small. It's worthless.
Okay, so very briefly about the classifiers we used. We started with just a rule-based classifier, just keywords. It's like the lowest line we definitely wanted to improve on. Then tried some classic machine learning classifiers in a binary relevance approach, so just training a different classifier for each separate label. Instead of, if you have one model, then it can take into account the correlations between labels, but the easiest way is just to have a separate model for each. Logistic regression turned out to be the best, like a classic algorithm, better than SVM. Then I tried a classifier chain, which is basically the same, was also logistic regression, but you first train the first label, and then when you do the next one, you feed in the true labels of the first label to the next model, and so on. And so that way the model can learn a bit about correlations. There's some downsides as well, because if you have a bad inference all the way at the start, it will propagate throughout your model, so actually perform much less well than the other ones. The current state of the art, as Charles already mentioned, is some kind of transformer model with self-attention, so basically all the LLMs are of that kind. I tried DistilBERT and SciBERT. SciBERT is just regular BERT. It's quite a big model, the same as the one that Charles used basically, but then with a scientific vocabulary. Not really taxonomy, but biomedical sciences, so should improve a bit. DistilBERT is much smaller, so fewer parameters, but a lot faster and requires less compute. We used it, so it's pre-trained on a very large corpus. We added a classification head that we tuned from scratch, and then we also fine-tuned a couple of the transformer layers themselves.
It's pre-trained?
So the BERT is pre-trained on So it's a lot of Wikipedia and so on, but also
Regular Bird is Wikipedia and Reddit, and a bunch of it's just random text from the internet. And then Cybert takes that and adds, I think it was pre-trained against like the entire PubMed open access corpus. So oodles of biomedical journal article text. Which at least just means hopefully, right, the hope is that it won't, you know, the one worry with transformer models is if a transformer model sees a word it's never seen before, right, and just doesn't know what to do with it because it can't correlate it with anything. So if you're hoping you've expanded its vocabulary enough that when you hit it with, now, it also got Wikipedia, so I mean, it's seen a bunch of science, right? But the hope is that Cybert's even a little better. That's the idea.
Yeah, and it is generally better for scientific, like text stuff. It does really do very well. But just for comparison, we also tried GPT. So just like we didn't give it one piece of text and then asked for the 40 labels because that didn't work at all. But we just asked like label by label, paragraph by paragraph, without any examples. There were several challenges in the whole thing. Most importantly, that we had very few samples. Definitely BERT works very well with a lot of data, but not very well with small training sets. Another big problem was that we had some categories that are very common, like phenotypic methods is basically 99% of the corpus, and other categories were like maybe 0.5% of the corpus, which is very tricky because your model will just optimize for the common categories and it will not care about the small ones. Usually there's tricks to solve this, but because this is multi-label and typically the sparse ones were combined with very common ones, once you start oversampling, you're going to also bias the common labels. So there was no very easy solution to deal with that. And then as always with the taxonomy, is the very obscure language, even for PubMed. There's like all the body parts of all the organisms it will not know. And the compute. So I don't have a GPU. The transformer models really need a GPU to work fast enough, and we didn't want to buy any compute. Still, performance was okay, I think. So on that side of the slide is the F1 score. That's sort of an aggregate of the recall and the precision. It gives a pretty good idea of how well your model does. So recall is how many of the true positives your model indicates as positive, and precision is how many of the ones that your model says are positive are actually positive. And F1 is like it aggregates this with a punishment for a big difference between recall and precision. On that side of the slide, for the left-hand side, the heads are all the most common categories, like 30% most common labels. Medium is the middle category. Tail are the 30% with the lowest frequencies. And not so surprisingly, for the two models we trained, so Cybert and logistic regression in blue and orange, it was just the more data there was, the better the model performs. Not surprisingly, GPT doesn't have this problem because we didn't give it any data. So it does almost as well on the tail and medium categories as it does on the head category. Then here on the right-hand side is sort of the overall evaluation of the models. So macro and micro F1, it's not like the left-hand side is just each label separately. The right-hand side is all the labels together. Macro F1 gives a bit of a punishment for bad classification of labels that are less common. It just gives equal weight to all labels. So not surprisingly, it's a bit worse in micro F1. The main thing I think to notice here is that Cybert isn't really better than logistic regression, despite its having, what was it, 110 million parameters. That's just the lack of data, I think. A few examples of labels. So the two on the bottom are the phenotype-based methods and the molecular methods. there's just a lot of data. They were very easy. You can see that the labels are very small. This one, for example, it's a label for the use of nuclear DNA. You can see that the models we trained did pretty poorly, and GPT did very well, which makes sense because there's so many nuclear genes you can use. There's only so many in our training set, but GPT knows all of them. Same with other categories that are like quite vague and broad, like there's rank just, that's rank justification, the category for when taxonomies justify why they classify a species, a group as a species or as something else.
Are you saying that ChatGPT knows a lot of molecular biology? Is it what you're saying?
This seems correct, yes.
Yeah, so
Okay, I learned something.
it's incredibly good at it. So I checked the literature on this before, and in molecular biology it's really very good. There's no literature about taxonomy itself, so we weren't sure. So in taxonomy as well, it does quite well. The labels it does very badly on, like shape, which is the label for when taxonomists describe the shape of something they're talking about, or like biogeography. I think it's partially a problem with the labels as well. They're very vague categories, like with it's sort of a continuum, and the human rankers and GPT might have put the threshold somewhere different, because when I looked into the misclassifications, I always sort of understood why it made the decision that it made. So I think there's some bad labels in there as well. And this is the cheapest model. Like you couldn't pay the more expensive models. You can have models that are 1,000 times better than this one. But anyway, it's not. So this is just on the training set. We cannot use it on our entire corpus anyway, because that would be way too much. So what we wanted to get from this, so one of the things we wanted to get was an estimate of how often methods are used in taxonomy. But because the classifiers do so badly, definitely for some labels we can't just trust the inference of our model. So we tried to get a better estimate using another model. So we take when we trained and tested a model, we kept one set of data apart to test it on at the end, to get a real idea of real-world performance. And from there we get the true positive, the false positives, and so on. And then we use the model to predict on all the unseen texts in our corpus, to get the actual observed counts. And then we just used a model to basically model that process and estimate the true proportions on the basis of that. So we turned the observed true positives and so on into a true positive rate and a false positive rate, which was just a flat prior updated by the actual observations. Then we put a p-true in our model, so the actual real proportion of, say, molecular methods or the use of shape. Again with a flat prior, because we don't really know much about it. And once we have that, we can say that whatever the model predicts is the true positive rate times the actual true rate, plus how many true negatives there are times the false positive rate. That's just everything that the model calls, like a positive instance. And then you model it as a binomial, and you get the actual estimate of the true count, or you can get posterior. And those are the blue bars on this figure. So those are the estimated counts in our corpus. The error bars are like the highest density interval, 95%. And the red dots are what Cybert actually predicted. So you can see that sometimes it's okay, sometimes it's very far off. I think it's when recall and precision were very far apart. Like if your recall is very low, but your precision is high, you're always going to underestimate the real proportion, and the other way around. So just to say that we will use this methodology data in the analysis I'm going to talk about now, but you have to take it with a grain of salt, because for some categories it's very far off, and it's not really possible to say anything like this on the individual paper level. So this is sort of an aggregate thing. So whenever we talk about something like non-phylogenetic genotype methods, the model is probably quite far off, and you have to take it with a grain of salt, but at least this gives an idea of the uncertainty we're working with. Okay, so that was all the data assembly. Now we have, I think, a really nice data set, quite unique in philosophy of science. I don't know if any data set about methods, and then on top of that all the other information, so there's quite a bit we can do with it. So now we'll just go over a few questions that have interested us for a long time, and that we might be able to answer now. So one thing is this tension between molecular and morphological work. So taxonomy has a very long tradition, that in the late 90s, early 2000s, there was this molecular revolution with a lot of debate between taxonomists. One of the big ones was this barcoding debate, like can you use short mitochondrial DNA bits to do taxonomy or to recognize species. There was a pretty quick consensus, a very trivial consensus, that probably people should use both, that there is room for both, and of course more is better, we should try to integrate them and so on. The question then is, well, did this actually happen? Taxonomy is a very big field. People writing in these journals are not the ones that only like go into the field and collect new organisms. So we looked very quickly on, so we just got this data a week ago or something. We quickly looked at how methods changed over time. One thing that's quite interesting on this figure is that the use of only phenotypic data has remained quite stable and very substantial over the past 20 years. So clearly there is a big part of taxonomy that has nothing at all, or that doesn't use molecular methods at all.
So recall from the year graph, yeah, the left-hand edge and the right-hand edge are sketchy,
Yeah,
right? They're
so-
much lower numbers, so.
So yeah, I think don't read too much in the fact that the blue part is very low at the start. Anyway, so you can also see the red part. There's a lot that don't use any methods at all, so that don't have any methodology sections. And that's using methods in a very broad sense. So as soon as anyone has a methodology section, we say they're using methods, but often they're not describing any statistics or any real data analysis. So I also try to pull that out. That's this figure. You see the green part, those are the people that use any statistical analysis, basically. So all of this is just people who say, we measured this and this, the specimen is stored here, something like that. So it's only this bit that uses some kind of analysis. So this green bit is this whole figure. You can see that at the start there was a lot of only using phenotypic data for analysis, but that has been almost entirely replaced by analysis that also includes some molecular methods. So basically now when people are analyzing data, it's almost always molecular data, and perhaps a bit of phenotypic data, but methods like collecting a data set of characters, like phenotypic characters, and then doing cladistic analysis on that. That was quite prevalent at the start, but it doesn't exist anymore today, almost. So it's clear that there still is a large part of taxonomy that is just basically phenotypic descriptions of species. You might think that maybe that's
Cost related.
Yeah, that's cost related. Maybe it's in the West people do molecular taxonomy, and in the poorer countries it's only phenotypic taxonomy. But that's not entirely true. Like, this is just a random subsample of I think 10,000 papers. But but Europe is almost the greenest part. And there are some red specks throughout.
Sorry, what's green and what's red?
Sorry, yeah, so the green is the phenotypic methods and the red is both. There's almost no genotype only.
And this is the author's location or the location of the species?
This is the location of the species, sorry, of the main location of the study. So it's not the author location, but the main location of the study.
Right.
Yeah,
Because it could be a European researcher doing research in, say, India.
yeah, I have a figure of that later on.
Okay.
So here it's just the main study location.
Okay.
If you divide it for the entire data set by continent, you do see that in South America there's a lot less molecular research and also fewer papers that deal with nuclear DNA. So typically the cheap molecular research is mitochondrial DNA. And I think a lot of the molecular work in poorer countries is mitochondrial DNA based. So another hypothesis might be that this part of taxonomy that just does phenotypic work, it's not really by geographic regions, but it's again these taxonomic communities. And so I actually used Charles's topic model.
It's not totally useless.
Because there were some topics that were clearly, like
Yeah.
I looked at some of the papers, they're clearly just this bunch of people that always write papers to each other and that are about, say, Asian leaf litter frogs or a certain kind of sponge.
Did you see a correlation with high disagreement in these subcommittees?
Come back to that.
And
I
so,
would expect, yeah.
yeah, for sure. And so there are big differences between communities in how often they use molecular methods. So the Asian leaf litter frogs, it's more than half of the papers use some kind of a molecular method, while in the mites or the leaf hoppers, there's almost no molecular methods at all. So I think a lot is segregated by those things, but they're not so easy to get at. Definitely with our corpus, because we have rather big journals, and a lot of these communities have their own like society of the blah 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 journal, where a lot of the papers go.
What is none?
What is
Phenotype only, and then none.
No methods.
No method section.
Yeah.
And
No
no
method
method
section.
section, okay. Not that there's no methods.
Well,
No, it's just it's not in the paper.
yeah, they don't report.
So, yeah. That's weird.
Yeah, I mean, it typically is, if you look at the papers, it's really someone who did field work, they find two organisms that they've never seen before and they just give a description. There isn't a real method that's being used either.
But can you infer?
Because it's presumably
Method?
phenotype based. If it's,
Yeah.
Can you infer that it's like, well, they are definitely not talking about molecular methods and they are using, I don't know, body parts,
That's,
and then that can you do that? I don't know.
it's quite likely, but we have to look at those papers more because there's also like other kinds of publications in these journals. There's maybe someone died,
Yeah.
or maybe there's a debate, like a nomenclatural debate. Corrigienda, there's lots of things that are in that category.
Okay.
Another interesting one that Charles already mentioned is the north-south imbalance. So, as part of the CBD, they launched this global taxonomy initiative because presumably taxonomy has an important role to play in dealing with the biodiversity crisis, so they wanted to boost taxonomy. And in particular, they wanted to boost it in the mega-diverse parts of the world. So in the unlucky situation that all of the research money is in places where there aren't so many species. And so all the Western taxonomists, they keep splitting local species into smaller chunks, while in the tropics there's a lot of biodiversity we haven't looked at. So the aim was really to get more people from the West there, but also to boost local taxonomy capacity. there's some indication that this might have worked. There was this 2012 paper by Costello that saw a small increase in taxonomists in Latin America and in Asia. But then other people keep launching new papers saying that we urgently need more taxonomic capacity in these places. So it's not so clear whether it actually worked. And I think our data set can help us look at that, but like Peter already mentioned, it's quite tricky to operationalize. Like we have the author countries, but there's often a lot of authors, and so it's not so clear where to go from there. We have the study locations, but again, like sometimes there's like 800 in one paper, like spread around the world.
A paper that reviews the entirety of a group, right? That will say, you know, oh, there's this guy who lives here, and 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 you'll get, of course, the way that I was describing that analysis, you get all of those.
So for now, I've just used the most common one. I've tried to control a bit for this issue of getting affiliations out of there by, like, if there were ties, I looked at the author countries and I took the other one. But we probably have to think about some kind of use I don't know, proportions or something like that. So we have to think about operationalization here. So a random subsample of papers for 4 time bins. Visually, there doesn't seem to be a big difference. Like ideally, we'd like to see like a band of red dots around the equator. We don't really see that. If you look at the proportions by continent over time, it's basically all the same except for Asia, which did increase. And I ran a regression as well, and that told basically the same story. So in Asia, clearly there's an increase.
We've had some other indications from other data sets that that's partly related to the development of the Chinese and Indian scientific communities, right? There's just lots of good taxonomists in China and India now,
Yeah.
Or lots more than there used to be, for sure.
We also looked at overlap with biodiversity hotspots as recognized by the Convention on Biological Diversity. And again, we had hoped to see a trend going up there, but there doesn't seem to be anything happening. I should say like the entire taxonomic capacity goes up rather strongly over time. So clearly there is more work in biodiversity hotspots, but proportionally there's no real change. So there's no shift of taxonomic effort from places that don't really need it to places that need it more urgently. This is the figure. So the red dots are the study locations and the blue crosses are the author countries. And so I just plotted it because I wanted to see whether there's a lot of blue crosses in the North America and Europe basically, and a lot of red dots in the south.
I didn't notice this until I was in front of the giant screen, by the way, but we got to figure out why the author country thing really hates France.
Really hates France?
Oh yeah. They just don't publish in English.
Or maybe it's the French, but yeah, something's... Well, no, because one of these journals is, the European Journal for Taxonomy is published by the Museum d'Histoire Naturelle. So, I mean, you think... Anyway,
in South
parentheses, that's
In
weird.
South America, I don't think that it's not hitting, that looks like Peru, no?
Yeah.
Bolivia? Oh,
In Bolivia.
in Bolivia, yeah.
Yeah, it's some problem with the data for sure.
Yeah.
How is the precise location of the blue dot?
Yeah, so for that, because we only have the author country, so I just made it
because
choose a random
not
point in
Russia
the
and Canada,
country.
clearly it's
They're not actually up there, yeah.
No, just random
all
points.
south of it.
But the study location...
Siberian taxonomy community is not that strong.
Oh yeah, maybe it would be interesting
But yeah.
to weight it over the population.
Yeah,
True,
that's
true.
We could do that. But yeah, Africa clearly has fewer authors than it has studies. It's basically only South Africa that has some taxonomic capacity. Then another long-running problem in taxonomy is bias towards certain kinds of biomes. Basically, all of biodiversity research focuses on forests a lot. Partially, I think, because forests are just a fun biome to study. But also, again, the same bias, like a lot of rich people live near temperate forests, and all the biodiversity there has to hide in the forest because there's not much left else. But it's not so easy to investigate this because you have to control, because forests do have a lot of biodiversity in general if you compare it to grasslands or something. So you would expect people to go there more often. And the area is also important. So when we just look at the raw counts, it is clearly true that forests dominate our data set. And then if we look at it by species share, so controlling for species richness, this basically is a proportion of 1 to 1, so anything below that is too low and anything above that is too high. You can see that it's mostly the temperate broadleaf and mixed forests, so it's basically Western Europe and North America, I think. And that the tropical and subtropical moist broadleaf forests might even be understudied, even though in absolute numbers they get a lot of attention. But this one, we'll have to think more carefully about how exactly to do it, because now I estimated the species richness of each biome by counts from vascular plants and vertebrates, which might not be so representative, but it's also very tricky to do it any other way. So I have to think about this.
Okay, let me be quick and wrap up. Just say, as I said, also, so we're interested in this question about taxonomic disagreement. What are those variables that we found might drive disagreement? The first one comes back to the question. So I instead of using the topics here, did something broader scale. So dividing up those species mentions into these kind of big chunky groups, like colloquially named groups, mammals, birds, mollusks, et cetera, et cetera, et cetera. Big categories. There is a lot more disagreement in bird taxonomy. That was actually quite expected. That's a very well attested fact. Ornithologists are really snippy. A bit more surprising is mollusks. When I first saw that number come in, I was like, well, that has to be an error from small n, but actually it's not small n. There's 1,000 mollusk papers, and they do have significantly more disagreement than others. You can read down the rest of the slide. There's actually not that many places that are notable for less disagreement. You can see prokaryotes here, but that's all. That is a signal of n equals 13, so that's garbage. So that's already kind of interesting. Might potentially be meaningful. I think cooler and a bit more interesting, a bit more meaningful, has to do with the age of the group being described. So I mentioned when we ID species, we can link them out to databases. Those databases, at least many of them, include a reference to the canonical place where the group was introduced, and you can get the date on the paper that was published. And so you can correlate between the year of the publication of the article that introduced the group, or in some cases it's literally like Linnaeus's book, right? And the disagreement index. Obviously, we're hypothesizing a negative correlation, right? You'd expect that the older the group is, the more you have to fight about it because it's already been described. And actually, it's exactly what you find, which is cool. It's a significant negative correlation. Around the mean value is about what you'd expect the difference to be between so you expect a paper discussing a Linnaean genus to have about 0.003 more disagreement than one describing a new genus, and 0.003 is pretty much the mean disagreeableness index value. So that's pretty hefty. That's a decent amount of explanation, I think. I've had a crack into this a little bit more. Yeah.
What's the variance of the disagreement index like?
I had this, but I don't have it in the slides. Okay. I could dig it off my laptop, but large.
Yeah.
I mean, disagreement is very, 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 very often zero. It is not uniformly distributed, which actually probably means that straightforward correlation is the wrong statistical test. But, or just a regression is the wrong, is not the most apt statistical test because it's not uniformly distributed. But at least this gives a basic idea that this is, I think this is going to be an important factor as we expected. And that's it. So yeah, questions, thoughts? We ran actually. That was almost exactly an hour. Yeah, we can do it. Let's do it. Let's do a brief break just to get water and stuff. I can use a bottle of water. Go set this RV load over here. Questions, thoughts, worries, immediate desire for beer? Yeah.
Sorry, so now that you have this tool, this data set, and these correlations, and these signals sometimes of non-linearity of correlations, complex stuff. So what is the next step to get from causal explanation? You use that method, you get more disagreement or whatever, something with the direction.
Yeah, I mean, you've got to supplement this with close reading at some point, right? And I mean, it seems like that's where, I mean, part of what I am hoping that this will give us, right, is indications about where close reading will be useful, where to go to either engage with the papers or in some cases engage with the practitioners, right? I mean, we can also go talk to taxonomists.
Yeah, or design, like or design other studies or experiments, because to really go at the causes, I don't think this data set it works. Also because we will do exploratory analysis now, which sort of disqualifies it from proper causal studies. So I think, yeah, really see if we can find anything here and then use that to properly inform other studies is the right way to go.
It seems that you have enough fine grain to get some proposition of difference makers. So not at the beginning of a causal study, you know.
I suppose we could do some like pseudo-causal stuff, like differences in differences or things like that, like not real causal designs, but sort of causal designs.
Clues for where to go.
If we get lucky and the Bayes net has the right shape, maybe we can infer a cause.
But at least some proposition where to go to.
Exactly. I mean, I think that for me, that's always been the hope. Excuse me. The hope has been. We knew about the existence of all these variables from the very beginning of the project, but then you look at that giant list of variables, and you're just like, well, what do you do now, right? How could you possibly? And so, finding the combinations that are maybe interesting to pursue further. Especially because we've had some, I mean, Stein's done some vignette studies in another part of the project where kind of, right, the naive thoughts about what were the important variables turned out not to be the important variables, actually, or at least weren't shown to be important by a study where you went and, proposed to taxonomists, hey, would this change your opinion if you did this way instead of that way? And it turns out, well, the answer was no. So hopefully, if we're driven by the literature, we will be able to pick out.
Yeah. So we had a lot of. So we started with a list of hypotheses that we wanted to look at, but those were sort of very strongly biased by what's very visible in taxonomy. Like they're very biased by all the bird stuff and the vertebrates, because there's lots of disagreement and there's like lots of certain social dynamics happening, and we hoped that we could look at those things with this data set. And now it turns out that actually 90% of taxonomy is somebody looking at mites and just giving a 5-line description of that mite. And what we see of taxonomy is basically the 0.1% of like very busy research communities that are not representative of what most of taxonomy is like. So in that sense, like all the disagreement stuff, and we had a lot of hypotheses and questions about the role that values played, which we can look at that partially because we have sort of for every paper we have what is it called, the SDG goals that are that figure in it. But again, it's such a small proportion that probably in 99% of taxonomy there is nothing value related at all, and so those hypotheses and questions become less interesting if we really want to say something about how taxonomy functions.
Are you able to map amateurs versus professionals in some way? That's my first question. The second question, the thing about the no methods, like you say, it feels like, do you think you will be able to pull apart a bit more sort of what's exactly going on there? Is it that they are really have no methods apparent, or that like you said, it's different kind of articles where it's not about describing a species? Do you think they will be able to pull apart a bit more to have a bit more like better data?
So the amateurs, there are some studies that do it, but those are really studies that just look at every paper, at every name, and look at who that person is. And it's typically people from the community itself who do that, who know 90% of all the people writing the papers. For us, like I think Charles has a model that like, cuts off all the affiliation stuff and so on, so we have that data, but to go from there to knowing who's at the university or not,
But there are some countries that have database of researcher in time. Not a lot, but there's countries that have that.
but that's probably personal data that people
But-
can just use.
They are used by Scientometric to do report about, when you blah, 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 and you invest, does it favor gender, blah, blah, blah. And they need
Yeah.
that kind of database to connect to the papers.
In the
Affiliation, gender, minorities, so they have that kind, they exist.
In the really nicely encoded, like XML encoded papers, we probably could pretty easily write a heuristic thing to siphon off everyone who's at a university.
But
But
most of the amateurs are at the university as well. So
that's a lot of them wind up with a kind of with a university affiliation line anyway, because they'll go glom onto a lab.
But maybe if you connect to I don't know how these databases are details,
Yeah, I don't know either.
but they exist, at least for certain Western countries, in Scientometric
I mean,
centers.
even Web of Science has all the affiliations and all the people, and I mean that's all available. It's very hard to do anything with that data. There's so many problems like mapping names to the same person, and then knowing what to do with affiliation. It's incredibly messy.
But that's their job, they manage to clean the thing, that's what they do. Maybe not, after that it's correlated to your database, that's complicated.
Yeah. And
Because a Scientometric center for a country, writing reports, they just spend 6 months every year just cleaning data. And after that, do the report, buy new data, clean the data for 6 months. This is what they do.
Answer the second question real quick.
sorry.
No, I know Max has to run.
You have to run that. So I think for some of the papers we have like the errata and the corrigenda and so on, we already have a way of knowing what those are, and the editorials, and there was a substantial proportion. And then I looked at 100 papers that were in that category of like no method papers, just to see how good our method was of extracting them. And I think 5% of them had some kind of a method section. 95% was really not a description of research at all.
Okay.
So we miss out on 5%, and that was typically because of a very weird section title, or because it was just like 2 paragraphs without a section title at all. But yeah, on a corpus of, on like I think it's 17,000 papers, 5% is still something.
In a world of infinite money where we could just shovel the full text of all of these papers at ChatGPT, I'm actually now fairly confident that it would classify the no method papers correctly. But that is a world of very infinite money that I do not live in.
And just one last question, but then I really have to go. I'm wondering if you're able to address like myths in the discourse that taxonomists have about themselves, about like, it's a sort of a community plagued with disagreements. But I'm always wondering, is it more than in, I don't know, linguistics or in philosophy? Is there more disagreement? Like, why are they talking about it in that way? And whether you are able to also show that there's a lot of things where there's quite a lot of sort of conservation of certain classification that sort of throughout decades and hundreds of years it's been sort of solid. So I'm wondering if you're able to address some of those like myths about the values, for example, that usually don't play a role.
So the reason, or the commonly accepted reason for why there's so much disagreement is that on the one hand it's a very subjective exercise classifying species. There's often many ways to go, and there's no one way. Like you can't just point at the data. And that combined with the fact that for a lot of these taxonomists, this is their hobby since they were 8 years old. They have their own taxon that they're really passionate about and that they go collect in their free time, and then they describe a new species and they're very emotionally attached to their particular classification. So this subjectiveness combined with like real emotional attachment, I think makes for disagreements. But yeah, the myths, that's actually what the questions we had in the final part of the talk, like the north-south and so on, that's what all the opinion papers are about. Like, we need more integrative taxonomy, or we need more taxonomists in the south. They're never based on data. They're always based on people's experiences of the field. So.
Same with the disagreement. One thing I haven't done yet, but that's on my very short-term to-do list, is just to look at the distribution of the disagreement index, right? I mean, you could say that it's a disagreement-heavy field, but if it turns out that there's only and I'm pretty sure this is the case, that there's a fairly small number of proportion of papers that actually ever talk in an article about disagreeing. Well, maybe you could have a very nuanced understanding where, well, everybody knows that it's disagreeing if it's a paper that describes the taxon in a different way than Smith did, and Smith's paper just came out last year, and maybe you could have that kind of a nuanced story, but I mean, it does start to crack the myth a little bit, right? If it just turns out that maybe they don't argue as much as we thought they did. But they definitely, you're right, they definitely, the self-image is, it's a field with a very powerful self-image, I feel like. I mean, whenever I talk to taxonomists, even evolutionary ecological biologists that don't directly do taxonomy, it's a very widely shared idea. Run away, run to the woods. Go ahead. It was like 3 simultaneous hands I saw.
No, but the younger people have priority.
Okay.
Go ahead.
So the question is about when you checked whether it's true that all molecular biology is done in rich countries. What was the precise methodology? Like you said, many articles have many authors. Was it like at least one author in this country? Was it like most of the authors from this country? Was it like all authors from this country?
So we looked at. Do we look at the author country, or do we look at the study country? And we used the most frequent one for that one.
Okay. Because I was thinking that maybe, I think it's quite a common thing that there will be, 5 authors and there will be 3 dudes from, let's say Nigeria, and 2 guys from, Germany, and the case will be that they will collect their data and send it to Europe or to the US, and all the lab work will be done in the US. And I was curious whether, with your data set, you can try to see whether that's a phenomenon or not?
Well, I actually tried first looking only at the final author, because I thought that will be the PI, and the first 3 will be like the local.
Yeah, but in this case there will be 2 PIs, because there will be one lab somewhere else and one lab elsewhere, and usually it's a collaboration between 2 different labs, or like field biologists and lab, something like this.
Yeah, I only looked at the first author and last author, and that didn't change anything. But if there's multiple PIs, yeah.
I was thinking something like try to look
So
at
in taxonomy, there's a tradition of citing in a certain order, or it's just...
Yeah, the PI is at the end, and then the PhD
Okay,
student.
that's the norm in taxonomy.
Yeah, it's also, it's the standard scientific, yeah.
Sorry.
Yeah, I was just thinking whether, for example, if you select only papers with all authors from one country, something like this, and whether you will see more difference.
Yeah, that's a good idea actually. We can try that.
Because I'm afraid that in this case it will be just messed up with this lot of articles where it will be half from one country, half from another country.
Yeah, that's true. I was actually thinking of just using proportions,
Oh,
like
okay.
whenever there's multiple, just some.
Yeah, weighted less.
It's the perennial question in doing this kind of work, full counting versus weighted counting. It's like a constant worry.
It was the
I thought it was just outside of Peter and Alexander.
Yeah, but please go.
So this is not very related to what you presented, which is great. Or maybe no, I'll ask first a question that is really related to what you presented. So I was wondering, just out of curiosity, if you use ChatGPT, like how do you use it? Do you really use it like normal people use it with a prompt, or is it like really? Just concretely, could you explain how you?
It's through the API.
Yeah.
So like programmatically, and there is you give a system prompt, and you give like there's two parts to your prompt.
Yeah.
And in one part you give like the general information the model has to take into account, and then in the other you give the concrete question. So we gave in the system prompt, we told it what it had to be like imagine your taxonomy is blah blah blah, those things. And we gave it a description of the category we were interested in. And then we sort of explained how it had to think about that category, and then in the actual main prompt we just gave it a bit of text, and then it knew from the system prompt it had to output a 1 or a 0, and only a 1 or a 0.
And how sensitive was it to the way you prompted it? I mean, because that's quite high level kind of information that you put in there, how they need to think about things and so on. Like that's
Yeah, so I did 4 runs with different ones, and it was for almost everything always the same. So one thing that you can vary is how deterministic it is. Like there is this parameter you can put higher or lower, and if you put it higher it's more creative. I put it very low because I just wanted it to give the most likely answer every time basically. So I wanted to know how well it works as a classifier. So changing that parameter did change the results, but the description itself, not so much.
The big advantage of the API is the ability to write your own system prompt, right? Because with ChatGPT, you're using its, which is, you are a helpful assistant who will be cheery. Yeah. So that's,
you
that's the
completely
big.
overruled that part.
Yeah, that's what makes ChatGPT versus GPT-04 or 4.0. It's the ChatGPT brand system prompt. And so that's the big advantage of the API, is getting to tell it, okay, you are a taxonomist, you are an analysis tool, you will spit out the following kind of answer, and it actually.
So there's no censoring.
There is, of course.
Oh.
Yeah. It still has its guardrails that are baked into the model at a level much lower than that.
That's not via prompting then.
I think they put the guardrails back in the system, or at least big asterisk. I think the last system prompts that were leaked for most of these chat models are pretty old now. But I remember when system prompts were getting leaked 6 months ago, 9 months ago, a year ago, the guardrails were repeated in the system prompt. But I think also there's work now, the models are internally guardrailed. I'm pretty
For sure, yeah. Most of the system prompts are open now. Like a lot of the companies release the system prompts, but the guardrails are in the post-training. It's part of the model itself. It's the same in the ChatGPT as in the API models.
Thanks.
So I saw that you said that you did some linear regression test from time to time to check dependence of variables. Did you try test with others kind of statistics? Because just in case, did you have a guess that maybe it's weirder? It's weirder.
Yeah.
It's weirder, and I should try another curve.
I'll say at the end, I haven't done any other testing yet, but I have the intuition that because I think the distribution of the disagreement index is going to be so far from normal.
Okay, so
This is probably the wrong. This is the wrong test. It's nice for a back of the envelope calculation, but For this, I think this is, I wouldn't publish this slide. I'm happy to talk about it, but I wouldn't publish
Okay.
this regression.
So you have to guess that some of your phenomena are really not.
Yeah, so for everything that has a count outcome, it was basically zero inflated models, like
Yeah.
negative binomial or Poisson, because 80% of the papers have zero, and
Yeah.
so you need to
Yeah.
model that separately. I tried splines on the continents.
You tried what?
Splines,
Okay.
like it's an easy way to get non-linearity, but it didn't do better in the model comparison, so I was just adding complexity. But I've looked at this data for a week, so there will be these were all just very quick tests to check intuitions, basically.
Okay.
Yeah, no, I mean, it's the classic problem that, as a rule, text features are power law distributed, right?
Well,
It's-
because I'm still dreaming about a causal model, so
Yeah, no, I mean,
maybe it's a dream.
we gotta get, step one is, this is a really nasty kind of, how to put it, I'm staring at a very similar problem as this in 3 different projects right now. It's a really nasty and challenging statistical problem when you have, quantitative and categorical data, and the quantitative data is distributed in a weird way, and you'd love to just hit a button and have the machine tell you like what things influence what other things. But man, I mean, this is how to put it. This is big boy statistics, which I'm not. Stan's way better at this than me. I'm not used to having to do good stuff.
Yeah, so the problem is we never have an idea of the causal graph at all. There's so many arrows everywhere. As long as you don't have an idea of such a graph, there's no statistics you can do. It will not make sense. So we hope to use this data to get such a graph, and then you can either test it with this, or you can design a test that will work. It's very messy.
Yeah, but that's new science. That's cool.
Yeah,
It's something we could not do before.
So for the vignette study that Charles described, we did have a very clear causal graph that we wanted to test, and then we can design a study, and that worked quite well. Sorry, there was a question.
Yeah, the question which Alexander actually you already asked, but I'm just curious about details. You said that it's an English language. How important is it? Like, I mean, if you take, for example, molecular biology journals and you limit yourself to English language, you don't lose anything basically. But clearly in field biology, probably you do. Is it taxonomy better than all other parts of field biology or not?
It's much worse. I think.
It's much worse.
Yeah.
Okay.
Much worse, yeah.
There's a bunch of for instance, one example that I know because I've seen it and I've flipped through some of the journals. There is a bunch of really good first order taxonomy of species in China written in Chinese. Huge journals, massive journals.
Russia and China are massive. But it's even for the taxonomists themselves, like they don't have access to this information. Sometimes they get, maybe now with the translation it's better, but all the taxonomists I talked to, they said that now and again they get a Russian colleague, and then when he's there, then everyone comes with their paper and asks him to read it and tell him what
Help!
happened. So,
Because I hope maybe that taxonomists will be more interested than general field biologists in having like a one big database. No?
Well, I mean, yes and no. I mean, there is a lot of momentum and scientific effort behind things like the GBIF and the Open Tree of Life, and et cetera, et cetera, et cetera, and smaller data sets too, like the marine worms database. I mean, there are some of these field-specific databases where, like, man, the practitioners are like obsessed with making sure that like the page about my worm in the marine worms database is like on point, right? So there are places where this matters, but I honestly don't know. I don't know what extent, those random first order taxonomists are really making sure that they're crunching their stuff out into the shared online databases.
So I know for the border of China, there's a whole like list of species that are described both in Chinese and in other language twice, like the same species.
Oh, sure.
There's like hundreds that have been described twice, because people just don't know.
Sorry.
No worries, see ya.
I think the main ones will be all the small societies with their own journal.
That's the other problem that I wanted to say something about. So an example that I always use here, because it deeply confused me at first, and then finally a biologist explained it to me. So one thing that I did way back in the beginning of the project to test whether we felt like the data set was representative is I sort of compared the species in the data set to the whole open tree of life, just to look at vague distribution, right? And you see some things you expect, like bacteria to go away, which is fine because bacterial taxonomy is a nightmare. But another thing that you saw that I didn't expect, that I was kind of confused and concerned about, is that the fungi went away. And a couple years into the project, I gave a talk actually here in Eli about this. And I showed that slide, and a guy came up to me afterward and he said, "I can explain your mycology problem." And I said, "How, really?" He said, "Yeah, no, even worse. So if you think the taxa you're looking at are bad, all mycology taxonomy work is published in the, Northeast German Friends of Mycology Society journal that's published out of some guy's basement in Bonn. And you're never going to get access to those papers. They're never going to be in your corpus." And so apparently, mycology is an outlier, but that's a really common phenomenon. I mean, that is a crazily common phenomenon, and I don't know, what can you do? There's always dark literature. Although here the dark literature is, there's a lot of dark literature.
Yeah, and you can get at them sometimes, but it's a lot of effort to get 312 papers, and then you have to do it again.
Out of that guy's basement, yeah.
For what concerns, for example, Chinese literature, at least concerns the name of species, don't they use the binomial Latin name? So in that case, it should be quite straightforward to include them into the search, right?
If you got the metadata, you could at least probably get major group name out of paper titles, for example. But then most of the rest of the data that we're trying to get out of these papers, we wouldn't be able to get to. So for some kinds of analysis, it might be useful. But yeah, I mean, partly too, part of what we're trying to do is one of the real advantages of the kind of setup that we have here, this kind of corpus, is that we've got enough data that we can do this wide variety of analyses on it. And so going backwards to titles and abstracts. Very often, especially now, since 2000, I think probably a majority now of the Chinese and Russian literature, there's a translated abstract that goes with most of those papers now too, right? That's put into the metadata. So that's accessible too. So a title and abstract data set would be feasible. But yeah, you'd lose a lot of the power that we are kind of excited to have.
Yeah, of course.
And one of the journals, so Taxa, has a lot of Russian and Chinese submissions as well. So it's not like they don't publish in the same journal either.
True.
Just a comment, I just found that the researcher that probably has a lot of database about other researchers to correlate to papers is Vincent Rivière, the UNESCO chair of open science.
Oh, Vincent,
Because he worked a lot on gender and minorities in science, so he had to build that. And I believe that probably the French Observatory of Science, the French thing that did the report, they must have a database of every damn researcher in France at least.
That has to be illegal for me to access,
But maybe you can ask. you know. It's a serious research.
sure. Yes. No, it would be interesting. I mean, this question about how to put it. I mean, that would be another. Speaking of this kind of self-image of taxonomists, I mean, another thing that would be it would be a crazily interesting variable to be able to mark every author as professional or amateur. I mean, there's all kinds of we could write multiple really interesting papers that taxonomists would be really excited to read if we could do that, because there's a lot of
or the mapping north-south that was
Right.
talking as that.
there's a lot of sort of self-introspection about like, oh, what do we do with the amateurs? What is this? This seems well, both this seems cool and is this okay in 2025? And yeah, I mean, I've read papers on both ends of that, and yeah, it would be really interesting to be able to do that.
Maybe it's all in the journals in the basement.
I mean, that's also, the dark literature problem probably does become much more serious, right? If you really want to do a concerted study of the amateurs. Yeah.
Yeah, I think there was a good European study that found that something like 42% of all new species over the past decades were by amateurs.
But they must be all with the website. You know the website where people put photos of everything they see in the forest?
Oh, iNaturalist.
High naturalist, and after that, I would not be surprised that there's a lot of new stuff coming just from this damn website.
Yeah, but to get a species described, you have to write, like you have to publish.
Of course.
And so on.
But at least the beginning of the process, and after that, it's written officially by someone. Because I think Axel.
Yes, he's obsessed with it, yes.
Axel is now co-author of a paper it's not a new species, but it's a new region where the species does not exist.
Oh.
And it starts from iNaturalist, and after that, they said, you know, in naturalist, you go to a step
know, he
higher,
actually got
and
the
people say, oh,
publication.
that species and that's location, that's not normal. And after that, someone write the papers.
I didn't know we got a publication out of it. That's
I don't know, he's prouder of that than anything he does here.
It's very rewarding. Yeah.
Yeah, I have also a question on, you also mentioned it, but the coverage of different taxa. Like, whether you should see, like you presented the stuff like forest versus non-forest, but inside different taxa, whether you see some sort of interesting results, or you're just afraid that you're that your data set is not representative. Because, for example, as you mentioned, there's like no precariat data, just 13 articles.
Right.
So, yeah.
Right. Super underrepresented in prokaryotes, super underrepresented in fungi, somewhat underrepresented in plants, overrepresented in vertebrates,
Yeah.
unsurprisingly. The rest looks pretty okay. The problem here is it's really hard. I mean, it's really hard to getting a good quantification of that representativeness is extremely difficult because there is no ground truth, right? I mean, the ground truth is the taxonomic literature.
Isn't your guess that the plants are published somewhere else?
Yeah, we only have one journal that does a lot of plants.
Yeah.
The other big plant journal we couldn't get. So.
Okay.
Yeah.
But we still have, like, we have an entire journal with only plants. So.
Yeah, so we have some plants. It's under. It's not that it goes away. It's less than you might hope for.
Yeah, and if you look at like taxon groups, like the categories Charles was talking about, like fish, birds, and so on, you see very big differences in like the use of certain methods or disagreement and things. So there's lots of patterns there. We didn't talk about them now, but there's some that really have splitting traditions. Like bird people, they keep splitting, or certain mammal groups, ungulates, they keep splitting. So you have a lot more disagreement there, you have a lot more papers with new species, while in other groups people keep lumping. So that gives different dynamics, and we can really see that in the data. So now we basically have to choose like which question or hypothesis that we had do we really want to go deeper into and do all the all the extra work that we need to do, because
Yeah.
there's really no-
The crunchy analysis work is yet to come, but we've got the tag data set and we're pretty confident in most of the tags. I need to polish up some of the disagreement stuff and polish up some of the location stuff, but otherwise we're pretty confident that we're not totally talking nonsense. Which, it took like 3 years to be confident that we were totally not talking nonsense. So I mean, that's already, I'm pretty happy that we've made it that far.
Okay, if there's no more questions, we can
Yeah,
call
Cool.
it a day.
call it a day.